Dear professors and dear competitor friends, first of all, hello everyone. This year the BioGate competition is being held for the first time. We are very happy to see you in this competition, and getting started first, I want to introduce ourselves. We are undergraduate students like you from Istanbul Technical University in Turkey, and 2 years ago we established the GALEN team, which consists of 35 students working for different fields of bioscience, especially on biotechnology, synthetic biology, and biosensor technology. We were also participating in different competitions, and we learned too many things from these competitions. One of the most important reasons for establishing this competition was to contribute to the development of students like you, and as much as we improve ourselves. For this reason, BioGate competitions consist of many different categories as synthetic biology, biosensor, artificial pandemic, computational biology, and astrobiology. And therefore, you have the chance to develop yourself in many different fields and to compete with many teams in international arena in this competition. We hope that you can broaden your mind when you develop your projects. And that's all, I think. Today, we have very valuable professors here who support us in the design phase of these categories and also advisor of BioGate competition. And Dr. Abdülhalim Kılıç is our advisor of team, and Dr. Behide Saltepe representing synthetic biology, Professor Dr. Gülay Korukluoğlu representing artificial pandemic, Professor Dr. Fethiye Aylin Sungur representing computational biology. And first of all, I want to start with Abdülhalim Kılıç, who is also the advisor of our team. He is the consultant of our team for about 2 years and the person who has helped us a lot. I want to leave the word to him without further ado.
Okay, thank you, Yaşar. I want to welcome everyone. I am Abdülhalim Kılıç. I am an assistant professor doctor at Istanbul Technical University at the Department of Molecular Biology and Genetics. And also I am, as Yaşar said, I am a supervisor of the GALEN team around 1 year, as I remember, if I am properly remember. So first, I want to welcome everyone, our supervisors for this competition, and welcome our students. I guess there are some students also from high school level, right, Yaşar? Probably.
Yeah,
I want to welcome everyone. It's a great pleasure for me today to make this opening speech also for this incredible initiative and this competition. This will be the first competition at the student level, actually, in our university, also in Turkey, within the under the synthetic biology category, actually. So I want to first appreciate you, Yaşar, your team for this organization, nice organization. I know very well that you deserve an appreciation because you actually performed a high effort to provide this organization in a suitable way, actually. And the BioGate competition actually starts with an motto, as it is indicated in the competition web page: the gate to the future, or the gate of the future. So actually, this competition really wants to intend to open a gateway to the future for these new generations, actually, which wants to intend to study or work in the field of biotechnology. So you know biotechnology and its concept, the concept from your lecture probably. So biotechnology is everywhere, and it has the potential or ability to transform whole world, right? We live in. So how we use biotechnology, we use biotechnology and tools, its methods to combat with the diseases, with pathogens. We try to develop our healthcare or enhance it, or how to say, we can revolutionize the agricultural methods, or we can preserve our environment from some pollutants, right? Or we can use some biotechnological tools for the food security. These are all very important aspects of the biotechnology. And you know that some developments required to actually transfer our humanity to the future. That's why the BioGate competition used this motto, a gateway to the future. So new generations actually, we need the new generations to be educated in this field. And I guess, I hope this kind of competitions will provide the nice environment and the suitable environment for the students actually to present themselves, present their projects, their ideas, and how to say, to make tight collaborations with the scientists, with other students, with innovators, with some companies, right? And it is just not a competition, of course. Please not just focus on to the winning group, so, or winning team, because another important point is to make or to feel the spirit of the collaboration or being a team, right? It is very important for the actually undergraduate or high school level students being an group actually, or being a team. So this kind of competitions provides platforms to gather, how to say, some creative or brilliant minds from the whole world. I hope the number is very high from the outside. I don't know, but I hope in the next years we will increase the number of the groups from the international students. But it will be a nice opportunity for you to show your creativity or to show your projects in the field of biotechnology. Of course, there are subclasses in the synthetic biology or biosensors or astrobiology, etc. Okay, so traditionally there are lots of congress or symposiums which are conducted at the academician level, right? But student congress or project congress are a bit different from this kind of symposiums, because at this level you show yourself, because as being undergraduate students, you have a bit different perspective from the academicians, because your I guess, I hope your curiosity or your creativity is forcing you, how to say, to challenge to conventional knowledge in the field, right? So we need this kind of ideas. So you will force the boundaries of the information or knowledge of the field. So that's why it will. I hope that BioGate competition will also provide you to show your skills, your passions, your creative ideas in the field of biotechnology, and I hope you can realize your project in the real life, real world. So I hope you can, you have the potential, you can show your potential to transform lives and, of course, create a better world for the next generations. So again, lastly, I want to again welcome you and thank you for your, how to say, contributions to these competitions. And I hope you all get excited throughout this competition. Again, thank you to the organizers, to team members, team captains, and our advisors already present here. And thank you again. Yaşa.
Thank you for your precious speech. And if it is okay, Behide Hocam, can you continue?
Yeah, let me share my screen. Can you see my screen now? Okay, I hope it's alive now. So first of all, welcome you all for the opening ceremony of the BioGate competition. And I would like to especially thank to the organizers for the invitation and offering me for being their supervisor on synthetic biology category. It is my pleasure to work with such dedicated and hardworking young researchers. My name is Behide, and I am working as postdoctoral researcher at the University of Edinburgh. And today I will give you a brief information about synthetic biology in terms of progress in time, applications, and limitations. Oops. So everyone has usually a different definition for the synthetic biology, but in general, it is the combination of biology with the engineering approaches to design new biological parts, devices, or systems, or redesign an existing system for functional improved purposes. So for this purpose, SynBio uses design, build, test, and learn cycle to build gene circuits, which are capable of different tasks. And for that, we have a pool of toolboxes to generate different types of gene circuits. Here is a summary of what you can do with the synthetic biology in general. So basically, you can produce an enzyme or chemical reliable for the industrial applications. You can design a cell which can detect and respond to a certain disease condition, or you can combat feedstock solutions issues, or you can battle for the environment. SynBio has some certain milestones, which I'm not going to that much detail, but in general, we see some specific time intervals throughout the last couple of decades. So the beginning of the field is around 60s, but the term of synthetic biology comes a bit later than that. So for the construction of the very first synthetic genetic circuits are around 2000, which are constructed by two different groups in US. With that, the foundation of synthetic biology has begun. And then with the iGEM competition coming into the picture, the expansion phase begins, and also within this period and afterwards, some more complex gene circuits have been proposed, and also more conceptual phenomena discussed, like standardization of the parts, modularity of the systems, using different assembly methods other than BioBrick assembly proposed in iGEM competition. And within this timeline around here, I came into the scene. So I got my bachelor degree from Istanbul Technical University, where I first met with synthetic biology and iGEM. So in 2013, we established the very first iGEM team in the university, which was composed of great people in our team. And then seeing all these opportunities in SynBio, I decided to pursue my PhD in Bilkent University. I mostly worked on engineered gene circuits for nanomaterial trigger toxicity detection, but also I contributed different projects during my PhD studies. And then as a dedicated biosensing person, I joined in the University of Edinburgh as a postdoctoral researcher. Now I'm working on wearable biosensors for non-invasive health monitoring. So to understand how synthetic gene circuits work, we need to understand how biosensors work in general. So basically, a biosensor is a device that can detect an incoming input and generates a measurable output. So basically, different types of recognition elements listed in here can detect a signal from the environment and transfer that signal into a measurable output. This output can be digital response corresponding to the on and off states of the analyte, or could be analog response corresponding to the concentration of this input within the environment. So cells are already evolved to understand the environment and react according to the changes, the fluctuations in the environment. So basically cells are already biosensors by themselves, which can be used to repurpose for different tasks. So this is the schematic of a cell showing how to understand and respond incoming signal from the environment. So in general, different types of inputs can be captured by transcription factors to component system or split DNA binding domains with the within the cell. And then these signals are transformed and processed by a signal processing unit, which is engineered gene circuits. Some examples are shown in here. And these circuits convert the incoming signal into a different types of output based on your application purpose. So for example, you can use, just for the sake of easy visualization, you can use fluorescent proteins for biodetection purposes, biosensing purposes, or you can produce a therapeutic agent to treatment of a disease. But using cells comes with a limitation of GMO regulations. So at this point, cell-free expression systems provides an alternative for cell-based applications. So basically, we are using the similar approach, but without the physical barrier of cells. And you can still use the similar machinery for the same purposes. In terms of field deployment, each area has its own limitations and considerations, but in general, they share some common grounds, like GMO considerations, long-term storage, stability, especially under harsh environmental conditions like low pH, or salt variations within the environment, temperature, and so on and so forth. But so far, some different, certain different strategies have been developed to bypass such limitations, such as conditional growth or kill switch mechanisms for cell-based systems, or using cell-free based expression systems to bypass the safety regulations. But one of the important development is to use physical containers like polymers, papers, or microfluidic devices to encapsulate either cells or the cell-free systems, which helps stability, long-term storage, and even easy visualization conditions in some certain applications. So with all challenges and solutions, I think synthetic biology holds a great opportunity in different fields, and researchers around the world are already working very hard to establish new ground rules for this in bio and extend its limitations to other fields as well. And I'm sure many more people will be interested in this field and start making some contributions in near future. With all due, I would like to wish a good luck all the teams who attending this competition, and thank you for your attention.
Thank you for your precious speech. Gülay Hocam.
Merhaba.
Hello.
Okay. Good evening, dear students and dear listeners. I would like to thanks to the organization committee for invite me very, very nice new approach competition. And when we met with the Galen team first time, when we talk about this competition, I was very excited, and it was so good news and good perspective for connection to the future. So first of all, I would like to introduce a little bit about myself, why I am here, and why I am the advisor as the artificial pandemic group. I am the microbiology. I am the medical doctor, and I am the microbiologist. Especially, I work in the virology area more than 28 years. When I finished my specialization program in Ankara, I started to work in Public Health General Directorate in Ministry of Health, which is related to the subject that we will talk about tonight. Because my department was the National Virology Laboratory, which is very important to monitor all the public health outbreaks. And because of the virus structure and the nature of the outbreaks, it is so important to detect such kind of the threat for the public health very early time. So in the National Virology Laboratory, we were searching all the some pathogens, some traits which can be effective on to our country's public health. So artificial pandemic is actually very, very important title for this competition, but as you know, we already had very huge pandemic within the last 3 years, and everybody and every sector was affected because of this pandemic. And then we should be very prepared. So I hope the young generation, as you and some students, will be interested. How can we make some solution? How can we offer some new approach? So I really give so much. I really emphasize the importance of this title within this perspective. And but also related to my issue, I can say for myself, I am the kind of the public health microbiologist, because any kind of the pandemic include a lot of different aspects, including the clinical sites, including the hospital management and patient management, including the antiviral therapeutics, and also the most important, one of the most important issue is vaccine development. So all these items will be investigated under the one umbrella. I hope you will be very supportive to think how can we manage better for better health, and how can we find the better solutions for the next coming future. And then, for example, in my situation, the WHO, World Health Organization, we already had such kind of the some exercise with the same title, artificial pandemic. But today I am very happy to do this with some young generation students, and I really very excited to learn your ideas, your solutions, your offers. So I wish to all of you very good, very successful competition area. Thank you so much for your joining us.
Thank you so much. Fethiye Hocam, you can start.
Okay. First, I would like to welcome my colleagues, candidate scientists, and enthusiastic participants. Actually, let me introduce myself first. I'm a professor at Istanbul Technical University, Computational Science and Engineering Department. My research interest is computational chemistry, biology. I mean, actually, it is not easy to separate them from each other. So modeling the reaction of biomicromolecules, specifically enzymes, ribosome, using molecular and quantum mechanics, computer-aided drug design are the subjects that I'm interested in. Before I start, I would also like to thank the organizers inviting me as a jury member to computational biology section. Dear organizing committee, keep your enthusiasm and amateur spirit alive. Thank you. For tonight, actually, they asked me to ignite the flames of inspiration for BioGate, specifically, of course, computational biology section. So in this context, I would like to share my experiences throughout the historical development of computational biology chemistry, together with the infinite possibilities of the area. Actually, in this age of exponential growth in technology and knowledge, we found ourselves at the forefront of a revolution that merges the power of computation with the complexities of life itself. It's an area where innovation and discovery are intertwined, and where our collective efforts can shape the future of the humanity. So, computational biology actually is the fusion of biology and computer science. Is not just a field of study. As Halim Hoca mentioned, for me, computational biology is a gateway to new frontiers where unravel the mysteries of life at the molecular level and also gain profound insights into mechanisms that governs life's most fundamental processes. Actually, it's a discipline that enables us to understand the complex network of genes, proteins, and cells and unlock their potential to revolutionize medicine, agriculture, and environmental sustainability. So in medicine, computational biology is a beacon of hope, actually. Why? Because it holds the key to understand complex diseases, and one may develop a personalized treatment. Through computational modeling and simulation, we can predict the effects of different drugs, identify potential targets for treatments, and accelerate the drug discovery processes. Imagine a world where a cancer is a conquerable enemy rather than a destructive one, where genetic disorders are detected and treated in advance. Why not? In this conjunction, actually, BioGate is not just a competition for me. I see it as a platform where the bright stars of the future in the biotechnology world come together. Why not? Speaking of the field of computational biology and chemistry, of course, I would like to mention the extraordinary achievements of Professor Ariel Worschel and Professor Martin Karplus, who were awarded the Nobel Prize in Chemistry for their pioneering work in computational modeling of enzymes. Actually, when you check our brochures, you will see that the main topic is the modeling of enzymes for the computational biology section. Their groundbreaking contributions have not only revolutionized the field of computational chemistry and biology, but have also changed our understanding of the building blocks of life and how biochemical reaction works. Through their innovative work, they developed a powerful molecular dynamics simulation tools. These tools allows us to visualize and analyze the motion of atoms and molecules in a very detailed way. By bridging the gap between theoretical calculations and experimental observations, actually they have given us a window into the hidden world of enzymes. At this stage, we must remember that computational biology is not a solo effort. It's a collaborative effort requiring the multidisciplinary teams where chemists, biologists, computer scientists, mathematicians, sometimes engineers come together to share their knowledge and expertise. And actually, we have to thank to the development of sophisticated computational algorithms and simulations, we can now observe the dynamic behavior of biological micromolecules, such as enzymes at the atomistic level. The unseen level of detail gives us invaluable insights into their structure, function, and the mechanisms for the catalysis of the chemical reactions. I should also mention about the drug design, because drug design can be approached through the both experimental and computational methods. Actually, drug design has gained great speed in recent years with the use of computational methods. The impact of the powerful combination of molecular dynamics and drug design reaches far beyond the laboratory walls. They offer hope to countless individuals struggling with diseases and conditions for which effective treatments are elusive. Why not? We can develop tailored therapies that combat cancer, infectious diseases, neurodegenerative disorders, and countless other health problems that affect humanity. So, using the power of computational modeling, we can screen vast libraries of compounds, predict their binding affinity to target enzymes or receptors, and guide the design of potent and specific drugs. This approach actually significantly accelerated the drug discovery process, and it enables us to identify promising candidates with less cost and time compared to the traditional trial and error approaches. Actually, at that point, BioGate, specifically computational biology section, offers all of you to show your talents and ideas to tackle real-world problems. As I conclude the remark, first of all, thank you all, and may the spirit of BioGate ignite the fires of inspiration within each of you. So let the competition begin. Good luck. Thank you.
Thank you for your amazing speech. Nurgül hocam, are you here?
Selam. Herkese iyi akşamlar. First of all, I sincerely apologize for my deep voice. I think I'm coming down with the cold. And well, I like to thank everyone for inviting me for such a nice competition. I'm in the committee of astrobiology. So this is as far as I know, it's going to be the first competition that is including astrobiology. I like to share a couple of slides if it's possible. If you give me the chance to share my screen, briefly explain why we are deeply interested in Earth history and geomicrobiology, what we can learn from this interdisciplinary science if we're interested in astrobiology in 5 minutes. So I guess I can share my screen, right? Okay. Is it okay? Everyone can see?
Yes, we can see.
Okay, all right. I'm trying to, in fact, actually, I'm trying to see myself. Something wrong with my screen. Sorry about that. I think I'm going to continue. Just. Okay. Yeah. All right. So, why are we interested in extreme environments and what we can learn from in terms of astrobiology? So luckily, we have lots of extreme environments in Turkey, and their significance or awareness about the extreme environment in Turkey is getting increasing, simply because of the searching for life in the universe. Primarily rely on what we understand from our home, Earth, and what extreme environments holding information in terms of not only how life started on Earth, but also beside Earth, such as Mars. So as the geomicrobiologist, we have the honor to direct and founder of geomicrobiology laboratory at ITU. As far as I know, this is the first and probably the only geomicrobiology laboratory in Turkey. So over the years, we have so much collaboration with different countries, and we are still continue to this international collaboration to get more information. Simply, what are some alternative Earths where life might existed. So what we try to understand is we try to understand the Earth history so that we can learn something about the origin and as well as evolution of life. In other words, we try to understand the alternative Earths throughout the history, because we know that the Earth now we are in, it's not like that. So it was hot. It was in some history it was cold, and as a result of this, it started the life evolve with the simultaneous changing lithosphere and biosphere. So if we understand these key parameters on Earth, then we can understand where life might be possible, or in other words, where the hot spots in our universe to look for life. But to do that, we know the biosignature that we interested in, the signals of life, in other words, on Earth, over time, provide the best guide in the search for life to recognizing biosignature somewhere else. In other words, studying the chemical signature of biological processes in our modern environments and the way they are captured in the geological records, our best tool. So again, we need to understand the geology, try to understand the sign of life, because all we have at the end, the rock that capture the sign of life, could be chemical, or could be biological, or isotopic records. So lesson from our planet's past and an alternative version of the Earth is our key. So we know that there's like milestone of life, and there has been many obstacle over the way, we should say. The first sign of life, it's come from 3.7 or 3.8 billion years, which is preserved in the stromatolite, and it's certainly there's a compelling evidence that this could be a cyanobacteria sign. So the cyanobacteria is the first good microfossils that preserve in the rock, which is stromatolite microbiota. We try to understand this microfossil signature on the rocks. So later on, Earth biospheres change shortly, and rock records recorded these changes. And then we know that the animals, 0.6 billion years, start to rise in the on the Earth, and the geochemistry is changed as a result of this biosphere-lithosphere interaction and atmosphere. Oxygen start accumulating in the atmosphere. So I'm trying to give you an example why we need to understand the geology and where it stands out in the astrobiology. So, right, again, these are some examples from the stromatolite, which is organosedimentary structure. They are, it's the biosignature, full of biosignature. That's why we specifically interested in this rocks in our lab. So we all, as astrobiologists, or myself, astrogeobiologist, we're hunting for life. So we need to understand the signature of life precisely, preserving the rock record. And this is not such an easy task, because on the screen, you see the cyanobacteria microfossil preserved in the Warrawoona, Australia, the rock. But on your right-hand side, you also see some optical micrograph of silica carbonate. It's purely made in chemical reactions. So you can see the incredible similarity, not only for morphology, And there's also the chemical similarities. This makes our job really difficult. That's why we need the modern environment on Earth to study in detail, to extend our knowledge of life, and then apply to exobiology studies. So Turkey is incredibly rich in this astrobiological side. For example, ultramorphic terrains, lakes with wide range of geochemistry, extreme environment, saline, alkaline, and hot springs, and the Black Sea, which is the largest anoxic basin in the world. So especially Black Sea, it's provide us national laboratory to study the life under anoxic conditions and full of H2S. In Lake Salda, which I have been working with for a long time, it's a good place and one of the best analog place on Jezero, the Paleo Lake, and Jezero Crater. And we keep continue and did this working with this. And in fact, we were able to create very nice analog places and look for biosignature preservation. And you can see some of the organic materials taken from the Lake Salda, and each layer that you see here, they have different microbial community profile. And as long as we can get information and microbial diversity of these different layers, we can apply to samples, presumably coming from Mars on 2030 to compare these results. Again, this is the fossil ones. It's not actively growing, but still may have some geological structure preserved in their rock record. And this is usually is a good indication of biology, although biology is not there. So this is the deep into detailed chemistry of some of the microbiological produce carbonate structure. Some of the fossil stromatolite, and you can see the biosignature on them. Some of the, over the years, we also work with the different hypersaline lakes in Turkey, and we know that some of the moons and terrestrial bodies have extremely salty environments. We presume that they're extremely salty, and then working with this salty anoxic environment, it gives us a clue to understand the initial condition and or microbial diversity of the lake. And these are the couple of examples that I like to share with you, just to give you an idea why the astrobiology it's a really standing as a key point for this this type of studies. And BioGate is it's really a nice competition, staying in the middle of each scientific division which have been presented here, that we can see that you need the chemistry, you need the physics, and you need the geology to make the right pathway. So I was really honored the part of this competition and as the committee member for the astrobiology. And I wish you all the luck. And as the representative of the geomicrobiology lab at ITU, I'm also honored to be share our knowledge and lab condition for the competition. And thank you again for invitation. And I'm really sorry for this deep voice. And I'll see you, I guess, in August, not even, maybe even earlier. And again, good luck for everything.
Dear Nurgül Hocam, thank you for your precious speech. And dear professors, thank you all for your amazing speech. I know that me and my friends will learn too much things from you during this competition. I hope that competitors also develop themselves in here. And I hope that they will discover themselves and even their theoretical parts I know that they achieve many important things in this competition. This year, we started an école, and I hope that this platform will continue to grow on. And maybe in a few years, this competition will reach thousands of students who inspired from here. And I'm very excited about to say that from now on, our competition has officially started. And you can reach us anytime you want in biogate@edu.tr. And we are waiting you on Istanbul at 19 and 20 August. Stay with science. Thank you for your part.